This is a presentation on the digestive system. So digestion is the mechanical and chemical breakdown of foods into forms that the cell membranes can absorb. And that's very important because they can only absorb those nutrients in a certain way, in a certain chemical. But the mechanical digestion is important also. It breaks down large pieces of food into smaller ones. Doesn't change the chemical composition. Chemical digestion, that's when we use the enzymes to break down the food particles. And what we do is change them into simpler chemicals that can be absorbed ultimately. But you got to break them into smaller pieces first so the enzymes can work on them. The digestive system includes organs that carry out mechanical and chemical digestion, but include the functions of ingestion, propulsion, absorption, and defecation. The digestive system consists of the alimentary canal and all the accessory organs. The digestive system consists of the two portions which we just mentioned. The alimentary canal is basically the food passageway. It consists of organs that extend from the mouth to the anus. So we're talking about the mouth, the pharynx, esophagus, stomach, small intestine, large intestine, and the anal canal. And we're basically talking about a muscular tube. It's about 8 meters long, so it's pretty long. Accessory organs include those that empty secretions into the alimentary canal. Now, food does not pass through the accessory organs, but we're talking about the salivary glands, the liver, the gallbladder, and the pancreas. Okay, this figure shows you the organs of the digestive system and gives you some basic functions of each. And we will go through these in more detail. Figure 17.2 here shows you what we consider the alimentary canal, going from the esophagus, stomach, then to the small intestine and large intestine to the anus. The wall of the alimentary canal is composed of 4 layers. From the innermost to outermost, the layers are going to be the mucosa. Again, that's the innermost layer. It's a mucous membrane. It directly surrounds the lumen, or the passageway. It's folded in some areas to increase surface area. It absorbs dietary nutrients. It secretes mucus and enzymes. Then we have the submucosa. It's connective tissue that contains blood and lymphatic vessels and nerves and glands. It nourishes the cells. It transports the absorbed food molecules. And then the muscularis externa, the muscle tissue basically. It contains circular and longitudinal layers, and it moves the tube and food materials. And then the serosa, that's the outermost layer. Serous fluid eliminates any friction. We're talking about the visceral peritoneum of the organs within the abdominal cavity. This shows you the layers of the wall of the alimentary canal in a little bit more detail. The mucosa is basically epithelium and connective tissue and some smooth muscle. Functions are for protection, secretion, and absorption. The submucosa, this is loose connective tissue, blood vessels, lymphatic vessels, nerves. It nourishes the surrounding tissues and transports the absorbed materials. Then the muscularis, this is the smooth muscle cells in the circular and longitudinal groups, movements of the tube and its contents or its function. And then we have the serosa. This is epithelium, connective tissue. It's for protection and lubrication. Okay, this figure shows you the structure of the wall of the small intestine and shows the 4 layers of the wall of the alimentary canal. There's 2 types of movement in the alimentary canal. There's the mixing movements. So the muscle in the small sections contracts rhythmically, does not move the materials in one direction. Examples, segmentation in the small intestine or churning in the stomach. And then you have propelling movements. These do move materials in one direction. Peristalsis is where you have the ring of contraction progressing down the tube. It propels food particles down the tract in a wave-like motion. And this figure shows you the movements of the tube. Okay, innervation. Branches of the sympathetic and parasympathetic divisions of the autonomic nervous system extensively innervate the alimentary canals. We have the submucosal plexus, which controls secretions, the myenteric plexus, which controls gastrointestinal motility, autonomic control of the digestive activity, includes the parasympathetic impulses, which increase the activities of the digestive system, so secretion and motility, and the sympathetic impulses. These inhibit digestive actions. So we're talking about inhibiting secretion and motility. And then we have the enteroendocrine cells. They're found in the stomach and small intestine. They secrete chemical messengers to regulate the GI organs and the processes. Okay, let's start at the mouth. This is the first part of the alimentary canal. You ingest foods there, of course, and then you have mastication, where you mechanically break down the solid particles and you mix that with saliva. And it functions as an organ of speech and sensory reception, surrounded by the lips, the cheeks, the tongue, and the palate, and includes the oral cavity and the vestibule. This figure shows you those areas of the mouth and the different parts. Okay, the cheeks. They form the lateral walls of the mouth. They contain the muscles for facial expression and chewing. They have an inner lining of stratified squamous epithelium, and it is moist epithelium. Then you have the lips, highly mobile structures that surround the mouth opening. It has sensory receptors that judge temperature and texture of food. The boundary between the skin and mucous membrane inside the mouth is included there. The tongue, the thick muscular organ that occupies the floor of the mouth and nearly fills the oral cavity when the mouth is closed. The lingual frenulum connects the tongue to the floor of the mouth. The papillae, these are the projections that move food and contain taste buds. And the lingual tonsils. This is lymphatic tissue masses on the root of the tongue. This figure shows you the tongue and the parts. The palate forms the roof of the oral cavity and consists of an anterior portion, the hard palate, and then a posterior portion, which is the soft palate. The hard bony palate consists of the palatine processes of the maxillae and the palatine bones. Soft palate consists of muscular arches that ends in the uvula. Palatine tonsils are the lymphatic masses on the sides of the tongue. And the pharyngeal tonsils, otherwise known as adenoids, are masses of lymphatic tissue in the posterior wall of the pharynx. This figure shows you the mouth, nasal cavity, and the pharynx, and those different parts. The teeth, these are the hardest structures in the body. They're not part of the skeletal system. Human dentition is that of an omnivore, eating all types of food. There's 20 primary teeth, or deciduous baby teeth, which are replaced by the secondary teeth. And then you have the 32 secondary or permanent adult teeth. The type of teeth include the central incisors, the lateral incisors, the canines or cuspids, the first premolars or bicuspids, the first molars, the second molars, and the third molars. They break food particles down to smaller pieces, beginning mechanical digestion, which increases the surface area for enzyme contact. Chewing mixes the food with the enzymes. The incisors are used for biting, the canines for grasping and tearing, the molars and premolars for grinding. And this consists of the crown and the root, which meet at the neck. The enamel covers the crown, The dentin makes up most of the tooth and surrounds the pulp cavity. The periodontal ligaments hold the root in place along with the cementum. This shows you the teeth of the child's skull. And you see that some of the permanent teeth or the secondary teeth are starting to come in and will push out those earlier teeth. And this shows you the teeth as well and the names of each of those parts. This table shows you the numbers of each of the types of teeth and their functions. And they also show you the age of the permanent tooth eruption. This shows you the section of a generalized tooth and the different parts, which we'll go over. Okay, clinical applications. The dental caries, cavities within the enamel of a tooth. They're formed when the sticky foods lodge between the teeth or in crevices of the molars. The bacteria on the teeth metabolize the sugars. The acidic byproducts then destroy the enamel and dentin. According to the American Dental Association, the following may help prevent dental caries: brush and floss regularly, regular dental exams and cleanings, fluoride treatments which strengthen the enamel, and sealants to prevent acids from remaining in the crevices and destroying the enamel. Okay, this table shows you the mouth parts and their functions and digestion. Okay, the salivary glands. They secrete saliva. They moisten the food particles and bind them together. It dissolves food so it can be tasted. It contains enzymes which begin the chemical digestion of carbohydrates and bicarbonate ions, which keep pH favorable for enzyme activity and protect the teeth from acidic foods. There's 3 pairs of major salivary glands. The parotid glands, these are the largest, and they secrete serous saliva with amylase. Submandibular glands at the floor of the mouth. They have serous plus mucus glands. And then sublingual glands, they're under the tongue and they're mainly mucus. Many minor glands are scattered throughout the mucosa of the tongue and the palate and the cheeks, and they keep the lining of the mouth moist. Okay, salivary secretions. The different pairs of salivary glands have varying proportions of 2 types of secretory cells. The serous cells, they produce a watery fluid containing a digestive enzyme called salivary amylase. Amylase splits starch and glycogen into disaccharides. The mucus cells, they secrete mucus. Mucus binds the food particles and lubricates the food while you swallow. Okay, table 17.4 shows you the major salivary glands and their locations, the ducts involved with them, and the types of secretions. This figure shows you the locations of the major salivary glands. This figure shows you light micrographs of the major salivary glands. So the parotid, the submandibular, and the sublingual glands. And as you see, the tissues are quite a bit different. And you'll see different types of cells in each. Okay, on to the pharynx and esophagus, working our way down. The pharynx and esophagus do not help in food digestion. Both the pharynx and esophagus have muscular walls which function in swallowing. The pharynx is a cavity posterior to the mouth. It extends from the nasal cavity to the esophagus. Parts of the pharynx include the nasopharynx, which is posterior to the nasal cavity, has air passages, contains openings to the auditory tubes. Then we have the oropharynx, which is posterior to the oral cavity, so air and food passage occurs here. And laryngopharynx is posterior to the larynx, and it's a passageway to The esophagus. The esophagus is a tubular organ that extends from the pharynx to the stomach. This figure shows you the muscles of the pharyngeal wall. As you can see, there are several. The swallowing mechanism. Swallowing can be divided into 3 stages. The first stage is voluntary. In this stage, saliva is mixed with chewed food, forming a bolus. In the second stage, this starts as the food reaches the oropharynx, stimulates the sensory receptors, and triggers swallowing reflex. The soft palate and uvula rise to protect the nasal cavity from food. The hyoid bone and larynx will elevate. The epiglottis closes off the top of the larynx to protect the trachea. The longitudinal muscles of the pharynx contract. The inferior constrictor muscles relax, and the esophagus opens. The peristaltic waves then force the food into the esophagus. In the third stage, peristalsis transports the food in the esophagus to the stomach. Okay, this figure shows you the different steps in the swallowing reflex. The esophagus. It's a muscular food passageway from the pharynx to the stomach. It's about 25 centimeters in length. It penetrates the diaphragm through the esophageal hiatus. It contains mucus glands in the submucosa, which moisten and lubricate the lining of the inner wall with the mucus. In the muscularis, this is the superior 1/3. This is skeletal. Then the middle part's 1/3 is skeletal plus smooth. And then the inferior 1/3 is smooth muscle. The lower esophageal or cardiac sphincter regulates food passage into the stomach and closes to prevent regurgitation of the food. These figures show you the esophagus, where it is, as well as a tissue cross-section. On to the stomach. So the stomach is a J-shaped pouch-like organ, which is about 25 to 30 centimeters in length. It's inferior to the diaphragm in the upper left portion of the abdominal cavity. Rugae are folds of mucosa and submucosa that allow for distension. The stomach receives food from the esophagus. It mixes food with the gastric juice. It initiates protein digestion, has limited absorption, but it moves food into the small intestine. It contains the following layers of smooth muscle: an inner circular layer, an outer longitudinal layer, and some portions have a third innermost layer of oblique fibers. Portions of the stomach include the cardia, the fundus, the body, and the pylorus. The cardia is the region near the opening to the esophagus, and it contains a lower esophageal sphincter. The fundus is a rounded area that rises above the cardia. It's a temporary food storage area which sometimes contains swallowed air. The body, this is the main portion. It lies between the fundus and the pylorus. And then the pylorus is the distal portion closest to the small intestine. Its funnel-shaped pyloric antrum narrows to become the pyloric canal. And the pyloric sphincter, this is a circular smooth muscle controlling the gastric emptying. This graphic shows you the parts, features, and the muscle layers of the stomach. Let's talk about the gastric secretions. Tubular gastric glands, which indent inward from the gastric pits, contain 4 types of secretory cells: mucous neck cells, chief cells, parietal cells, and enteroendocrine cells, which produce a mixture called gastric juice. Mucus, this provides lubrication and protects the stomach lining, secreted by the mucous neck cells. Pepsinogen, this is an inactive form of pepsin secreted by the chief cells. Pepsin, this is an active enzyme that breaks down proteins into polypeptides. It forms from pepsinogen in the presence of hydrochloric acid. Gastric lipase, this is a fat splitting enzyme found in the small quantities. Action is inhibited by low pH. The hydrochloric acid. This converts pepsinogen into pepsin and is produced by the parietal cells. Intrinsic factor. This is required for absorption of vitamin B12, and it's produced by the parietal cells. Chemical messengers. These are substances such as hormones and local messengers that regulate digestive processes. They're secreted by enteroendocrine cells. This table shows you the major components of gastric juice. Okay, so mucus provides an alkaline mucus that protects the stomach's inner surface from hydrochloric acid and enzymes. Pepsinogen is an inactive form of pepsin. Pepsin is a protein-splitting enzyme that digests nearly all types of dietary protein into polypeptides. Hydrochloric acid provides the acid environment needed for production and action of pepsin. The chemical messengers, they regulate digestive motility and secretion. And the intrinsic factor, it's necessary for vitamin B12 absorption into the small intestine. This figure shows you the lining of the stomach. Regulation of the gastric secretion. So gastric juice is produced continuously. The rate of production varies and is under neural and hormonal control. In the neural regulation, sympathetic impulses decrease the gastric activity. The parasympathetic impulses increase gastric activity and promote the release of histamine, which stimulates gastric secretion. Hormonal regulation includes somatostatin, which inhibits hydrochloric acid secretion, gastrin, which increases gastric juice secretion, and cholecystokinin, or CCK, which is released by the small intestine cells when the proteins and fats enter the small intestine. It decreases gastric motility. This figure shows you regulation of the gastric secretions. This table shows you the phases of gastric secretion. So in the cephalic phase, the sight, taste, smell, or thought of food will trigger parasympathetic reflexes. Gastric juice is then secreted in response. The gastric phase. Food in the stomach chemically and mechanically stimulates the release of gastrin, which in turn stimulates secretion of gastric juice. Reflex responses also stimulate gastric juice secretion. In the intestinal phase, as food enters the small intestine, it stimulates intestinal cells to release intestinal gastrin, which in turn briefly promotes the secretion of gastric juice from the stomach wall. This phase primarily inhibits gastric juice secretion by sympathetic reflex and the hormone cholecystokinin responses. Okay, in gastric absorption, the gastric enzyme pepsin begins breaking down the proteins. The wall of the stomach is not well adapted to absorb digestive products. Most nutrients are absorbed in the small intestines. The stomach absorbs these substances though, some water, certain salts, certain lipid-soluble drugs, and some alcohol. The mixing and emptying actions. The mixing of food in the stomach with gastric juice produces a semi-fluid paste called chyme. Peristaltic waves push the chyme towards the pylorus of the stomach. Small amounts of chyme are transported through the pyloric sphincter at a time. When peristaltic wave reaches the distal portion of the stomach, the contents are pushed backward, resulting in mixing action. Lower esophageal sphincter prevents the backflow of chyme into the esophagus. The rate of emptying depends on the consistency of the chyme and the type of food. As chyme enters the duodenum, accessory organs add secretions to act on the chyme. When chyme starts to fill the duodenum, stretch receptors initiate the enterogastric reflex. This slows the stomach emptying and intestinal filling. This figure shows you the stomach movements. This shows you the enterogastric reflex. Okay, common problem, heartburn. Stomachache results from eating a lot of food too quickly. It takes up to 20 minutes for the hypothalamus to sense a full stomach. Excess fullness leads to abdominal pain and gastric reflux as the stomach contents enters the esophagus. Stomach contents in the esophagus causes inflammation, called esophagitis. Feels like the pain is derived from the heart, so it's called heartburn. Antacids can provide some relief. For prevention, eat small meals, eat slowly, stay upright after eating, and avoid caffeine, nicotine, and alcohol. Vomiting. This is a result of the reflex that empties the stomach in reverse direction. It includes certain drugs, toxins from food, overstretching of the stomach, body motion changes, motion sickness associated with the inner ears. It's controlled by the vomiting center in the medulla oblongata. The motor response involves deep breaths, the raising of the soft palate, closing of the nasal cavity and glottis, relaxation of the lower esophageal sphincter, contraction of the diaphragm and abdominal wall muscles, and it forces the food out through the mouth. Okay, let's look at some of the associated organs. The pancreas. This is an endocrine gland secreting insulin and glucagon to regulate the blood glucose. Also, an exocrine gland of the digestive system. It secretes digestive fluid called pancreatic juices. The structure of the exocrine pancreas includes pancreatic acinar cells that make up most of the pancreas and release the pancreatic juice into tiny ducts, which lead to the pancreatic duct. The pancreatic duct, along with the common bile duct from the liver and gallbladder, empties into the duodenum of the small intestine. The pancreatic duct and the common bile duct join a dilated tube called the hepatopancreatic ampulla of the Vater. The hepatopancreatic sphincter of Oddi surrounds the ampulla and controls the movement of bile and pancreatic juice into the duodenum. This figure shows you the structure of the pancreas. Okay, the pancreatic juice contains enzymes that digest carbohydrates, fats, proteins, and nucleic acids. Components of the pancreatic juice include pancreatic amylase, which splits starch and glycogen into disaccharides, pancreatic lipase, which breaks down triglycerides, trypsin, which digests proteins, released as inactive trypsinogen, which is activated by enterokinase in the small intestine, chymotrypsin, which digests proteins. It's released as an inactive form, but it's activated by trypsin. Carboxypeptidase digests proteins. It's released as an inactive, but it's activated by trypsin. Nucleases, they digest nucleic acids. And bicarbonate ions make pancreatic juice alkaline and buffers the stomach acids. Regulation of pancreatic secretions includes the use of hormones that help regulate the release of the pancreatic juice. Secretin stimulates pancreas to release the pancreatic juice high in bicarbonated ions. Cholecystokinin stimulates the pancreas to release the pancreatic juice high in the enzymes. Nervous system regulation. This is during cephalic and the gastric phases. The parasympathetic impulses stimulate the pancreas to secrete digestive enzymes. This figure shows you the regulation of the pancreatic secretions. The liver and the gallbladder. The liver is the largest internal organ. It's located in the upper right abdominal quadrant, just beneath the diaphragm. It's a reddish-brown organ, and it's well supplied with blood vessels. The liver has 4 lobes. The right lobe is the largest. The left lobe is smaller. The quadrate lobe is a minor lobe near the gallbladder, and the caudate lobe is a minor lobe near the inferior vena cava. Lobes consist of lobules, which are the functional units of the liver. They contain hepatocytes or liver cells around a central vein. The hepatic sinusoids are channels for blood between the plates of cells. The hepatic portal vein brings absorbed nutrients to the sinusoids. The hepatic artery brings oxygen-rich blood, which mixes with the oxygen-poor blood from the portal vein. Blood flows from the sinusoids to the central veins, then to the hepatic veins. Bile flows from the ductules to the hepatic ductules, then to the common hepatic duct. Figure 17.5 shows you a transverse section of the abdomen, showing the liver, the gallbladder, and other organs. This figure shows you the lobes of the liver. This figure shows you the hepatic lobule structure, showing the arrangement and the triads. This figure shows you the paths of blood and bile in the hepatic lobules. All right, let's talk about the liver functions. The liver carries on many important activities. It produces glycogen from glucose. It breaks down glycogen into glucose, converts non-carbohydrates to glucose, it oxidizes fatty acids. It synthesizes lipoproteins, phospholipids, and cholesterol. It converts excess carbohydrates and proteins into fats, deaminates amino acids, it forms urea, it synthesizes the plasma proteins, it converts some amino acids to other amino acids, it stores glycogen, iron, and vitamins A, D, and B12. It phagocytizes damaged red blood cells and foreign substances and antigens. It removes toxins such as alcohol and certain drugs from the blood. It acts as a blood reservoir, and it has a role in digestion, which is to secrete bile. This table shows you those major functions of the liver with a little bit more detail. All right, replacing the liver. Well, sometimes the liver goes bad in individuals, and the liver is required to maintain life. The liver is capable of regeneration if 25 to 30% of it is healthy. If cancer spreads to the liver, life can continue only weeks to months. A donor can donate parts of the liver as regeneration can occur. There is a shortage of donor livers for transplant, though. Extracorporeal liver assisting device, or ELAD, can temporarily perform blood cleansing functions of the liver until a donor liver becomes available. ELAD is bioartificial, since it contains artificial portions like the cartridges and biological portions like human liver cells. The patient's blood plasma is passed through the ELAD, where toxins are removed and liver secretions are added. Then the plasma is returned to the patient. Bile is a yellowish-green liquid that hepatic cells continuously secrete. Components of bile include water and bile salts, which are produced from cholesterol. They emulsify fats, only bile components that have a digestive function, and they act as a fat emulsifying agent. Bile pigments include bilirubin and biliverdin, which are derived from hemoglobin breakdown. Then you also have cholesterol in bile and electrolytes. In jaundice, you get a yellowing of the skin and the eye sclera and the mucous membranes. They're caused by the blockage of bile ducts and diseases such as cirrhosis or hepatitis, or rapid red blood cell destruction. Hepatitis. This is an inflammation of the liver. Many types occur, chronic and acute, and types vary in severity. Some people have symptoms and some do not. They can still be carriers, though. At first, symptoms resemble the flu. Symptoms progress to a rash, pain in the right upper quadrant, dark urine, pale feces, and jaundice, which is yellowing of the skin and sclera of the eye. The most common cause of hepatitis is one of several types of viruses. Some forms are bloodborne. Some forms are transmitted by contact with food, body fluids, or objects contaminated with feces that contain the virus. Hepatitis A is transmitted by contact with food or objects contaminated with feces containing the virus. Hepatitis B is transmitted by contact with body fluids containing the virus. Hepatitis C is transmitted through blood contact. Half of all known cases of hepatitis. There are newer medications that are successfully treating hepatitis C. There are other forms such as hepatitis D, E, and G. Antibiotics are not effective against hepatitis because it's viral. There are effective vaccines, though, to prevent hepatitis A, B, and E. The gallbladder. This is a pear-shaped sac. It's found on the inferior surface of the liver. The cystic duct, which is the only entry and exit duct, merges with common hepatic duct to form the common bile duct. It stores and concentrates bile. Releases bile into the duodenum of the small intestine via the common bile duct at the hepatopancreatic ampulla. The hepatopancreatic sphincter regulates release of the bile into the duodenum. Gallstones. The gallbladder normally concentrates bile salts, bile pigments, and cholesterol. Sometimes the cholesterol precipitates and forms solid crystals. The crystals can enlarge, which form gallstones. The causes include excess bile concentration, too much cholesterol secretion by the liver, or inflammation of the gallbladder. Bile release is under control of the hormone cholecystokinin, which stimulates the contraction of the gallbladder. Figure 17.29 shows you a gallbladder with gallstones. This figure shows you regulation of bile release by cholecystokinin. Functions of bile salts: aid digestive enzymes through emulsification. You have the breakup of large fat globules into smaller droplets, like soap or detergent. They aid in fat digestion by increasing the surface area accessible to the enzyme lipase. It enhances absorption of fatty acids and cholesterol by forming micelles. Helps absorb fat-soluble vitamins A, D, E, and K. And almost all bile salts are recycled. Okay, gallbladder disease. Symptoms of gallbladder disease include pain in the right upper abdominal quadrant, the back and right shoulder, perhaps nausea and sweating. Tests to observe the gallbladder include ultrasonography of the gallbladder or an X-ray called a cholecystogram. Gallstones can be found in the gallbladder, cystic duct, hepatic duct, or the common bile duct. Treatments: cholecystectomy, which is a surgical removal of the gallbladder, endoscopy to find stones or remove them from certain areas. This table shows you hormones of the digestive tract and their functions. Now we move to the small intestine. It's a tubular organ that extends from the pyloric sphincter to the beginning of the large intestine. It fills most of the abdominal cavity. It receives chyme from the stomach and the liver and the pancreatic secretions. It completes digestion of the nutrients in chyme. It absorbs the products of digestion and transports the remaining residue to the large intestine. The small intestine consists of three parts: the duodenum, jejunum, and the ileum. The duodenum, also pronounced duodenum, is the shortest and most fixed portion of the small intestine. The jejunum is the middle portion. It's thicker and more active than the ileum. And the ileum is the distal portion that contains Peyer's patches, which are lymph nodules. Jejunum and ileum are suspended by a double-layered fold of peritoneum called mesentery, which transports blood and lymphatic vessels and nerves to the wall of the intestine. A double fold of peritoneum, called the greater omentum, drapes down from the stomach over the large intestine and folds of small intestine. This figure shows you parts of the small intestine. These figures show you the mesentery and the greater omentum. Structures of the small intestinal wall. Tiny projections of mucosa, called intestinal villi, greatly increase the surface area for absorption of digestive end products. Each villus consists of a simple columnar epithelium with a core of connective tissue, with blood vessels which absorb most of the nutrients, a lacteal, which is lymphatic capillary that absorbs large fats, and nerve fibers. The free surfaces of epithelial cells contain microvilli, which also increase surface areas for absorption. The intestinal glands, or crypts of Lieberkühn, are located between the bases of the villi. The plicae circularis are circular folds of mucosa, which also increase surface area. These figures show you the villi of the small intestine. This figure shows you the intestinal epithelium. These figures show you the plicae circularis, the villi, and layers of the wall of the small intestine. Secretions of the small intestine. Mucus, which is secreted by the goblet cells. It's also secreted by specialized Brunner's glands that secrete a thick alkaline mucus in response to certain stimuli. Watery fluid. This is secreted by the intestinal glands. It picks up digestion products and transports them into the villi. It does not contain digestive enzymes. Enzymes in the membrane of the microvilli. So peptidases, they break down peptides into amino acids. Sucrase, maltase, and lactase, which break down disaccharides into the monosaccharides. Lipases, which break down fats into fatty acids and glycerol. Chemical messengers, such as secretin and cholecystokinin. It's secreted by the enteroendocrine cells scattered throughout the mucosa of the duodenum. And antimicrobial substances that defend against certain bacteria. They're secreted by special cells in the intestinal glands. This table summarizes some of the major digestive enzymes. Regulation of the small intestinal secretions. So this occurs by several methods. Mucus secretion occurs in response to mechanical stimulation or the presence of irritants, such as gastric juice. Contact with chyme stimulates goblet cells to secrete mucus and intestinal glands to secrete watery fluid. Distension of the intestinal wall activates nerve plexuses in the wall of the small intestine. Distension also stimulates the parasympathetic reflexes that trigger the release of intestinal enzymes. Okay, absorption in the small intestine. Villi increase the surface area for absorption. The small intestine is the most important absorbent organ of the alimentary canal. Absorption is so effective that very little absorbable material reaches the organ's distal end. Carbohydrate digestion and absorption. Salivary and pancreatic amylase breaks down starch and glycogen into disaccharides. Intestinal enzymes break down disaccharides into monosaccharides. The monosaccharides are absorbed by facilitated diffusion and active transport into the blood vessels in the villi. Protein digestion and absorption. Pepsin in the stomach breaks down proteins into polypeptides. The pancreatic proteases, like trypsin and chymotrypsin, break down proteins and polypeptides into smaller peptides. The intestinal peptidases break down peptides into amino acids. And amino acids are absorbed by active transport into the blood vessels of the villi. This shows you the disaccharide and dipeptide digestion. Fat digestion and absorption. It's emulsified by bile salts. It's digested mainly by enzymes from the pancreas and the small intestine. It's digested into glycerol and fatty acids. And the fatty acids and glycerol are absorbed by a process involved several steps. Absorbed into the blood or the lymphatic capillaries, which are the lacteals. The micelles and chylomicrons. The micelles are loose complexes of fatty acids and bile salts, from which fatty acids can migrate to the microvilli and be absorbed. Fatty acids and glycerol are resynthesized into triglycerides in the smooth endoplasmic reticulum. New triglyceride clusters are encased in protein, forming chylomicrons, and chylomicrons are absorbed by the lacteals. This figure shows you fat digestion. This figure shows you triglyceride absorption. This table shows you intestinal absorption of nutrients. Movements of the small intestine. The small intestine carries on the following movements. Peristalsis. These are wave-like pushing movements that propel chyme in proper direction down the small intestine. Segmentation. These are ring-like contractions that move chyme back and forth, which is a mixing motion. It places chyme in contact with digestive enzymes in the brush border and aids in absorption. Parasympathetic impulses stimulate both mixing and peristaltic movements, and sympathetic impulses inhibit them. If the small intestine becomes overdistended or irritated, strong peristaltic rush may sweep contents quickly into the large intestine, resulting in diarrhea. Ileocecal sphincter joins the ileum of the small intestine to the cecum of the large intestine and helps regulate the flow of chyme. And down to the large intestine. It's named because of its diameter. It is greater than that of the small intestine. It's about 5 feet long. At the distal end, it opens to the outside of the body through the anus. It absorbs some water and electrolytes. It reabsorbs and recycles water and digestive secretions and forms and stores feces. Okay, parts of the large intestine. The cecum. This is a pouch that forms the beginning of the large intestine. The appendix is attached to the cecum. Lymph nodules in the appendix function in immune response. The colon. You have the ascending, transverse, and descending and sigmoid portions. They contain the hepatic right colic and the splenic left colic flexures. The rectum. It extends from the sigmoid colon to the anal canal and lies next to the sacrum. The anal canal, it goes from 2.5 to 4 centimeters of the large intestine and opens to the outside as the anus. The internal and external anal sphincters guard the anus. This figure shows you an anterior view of parts of the large intestine. These figures show you the large intestine, the rectum, and the anal canal. Okay, structure of the large intestinal wall. The wall has the same 4 layers as other tubular organs of the alimentary canal, but it does not have villi and plicae circularis. The longitudinal muscles are organized into 3 bands. The taenia coli that run down the entire length of the colon, and then the muscle bands create pouches called haustra, which help form feces. This shows you a micrograph of the large intestinal wall and the different tissues and layers. Okay, the large intestine has little or no digestive function. It contains tubular glands containing goblet cells that secrete mucus, the only significant secretion of the large intestine. It does absorb water, about 90% of the water that enters it, and electrolytes. It houses intestinal flora, or bacteria which break down the contents, such as cellulose, and that produces vitamins K, B12, and thiamine. It forms feces and carries out defecation. Intestinal gas is a mixture of nitrogen, oxygen, methane, carbon dioxide, hydrogen, ammonia, and hydrogen sulfide, which causes an unpleasant odor. This figure shows you the large intestinal mucosa. Okay, movements of the large intestine are similar to those of the small intestine. They're slower and less frequent than those of the small intestine. Types of movements include mixing movements, like mass movements, which are peristaltic, 2 to 3 times per day, which usually follow meals. and the defecation reflex, which eliminates feces from the body. It involves holding deep breath, contracting the abdominal muscles. Feces then move into the rectum. Peristaltic waves occur in the distending colon and relaxes the internal and sphincter and then the external anal sphincter. Feces are composed of materials not digested or absorbed and also contain water, that's about 75% of feces, electrolytes, mucus, bacteria, and bile pigments, which provide the color after the bacterial alteration. The pungent odor is produced by bacterial compounds, including phenol, hydrogen sulfide, indole, skatole, and ammonia. Okay, disorders of large intestine include diverticulosis. This is a weakening of the intestinal wall, which leads to protrusion of the mucous membrane. It forms outpouchings that can become inflamed and infected. Inflammatory bowel disease. This is a group of disorders including ulcerative colitis, which affects the mucosa and submucosa of large intestine and causes bloody diarrhea and cramps. Crohn's disease. This is more serious and affects all layers and occurs in both small and large intestines, causes diarrhea and pain. Colorectal cancer. This is cancer of the large intestine or rectum. It's the fourth most common cancer in the United States. Screening tests include fecal occult blood tests and colonoscopy. Lifespan changes for the digestive system. Changes to the digestive system are slow and slight and eventually include the following. Tooth enamel thins and teeth become more sensitive. The gums may recede. The teeth may loosen, break, or fall out. The GI tract becomes less efficient. Slowing peristalsis may lead to heartburn. Gastric secretion slows. Constipation may become more frequent. Nutrient absorption decreases, including fat-soluble vitamins. Incidence of lactose intolerance increases. And accessory organs age, but typically not necessarily in ways that affect health. And that is the end of our digestive system presentation.